All right, good afternoon, everybody. Afternoon. Let me close the door here. All right. How's everybody doing? Doing okay? Yeah, tired? How many people are tired this week? My hand's raised as well. Yeah, it's long. Yeah, midterms are maybe over now, but doesn't feel much different for this class. I know got a lot coming at you this week for sure. Yeah, definitely a lot going on. Yeah, and I was able to make it to chapel today. How many people were at chapel? Yeah. The speaker's been fantastic this week. Definitely enjoyed her. I thought the message, the final take home point today of the chill acronym was pretty good. This idea that if we have big emotions that come up for some reason, then maybe take a look at those, see what's going on, try to figure out what does God have to teach you, maybe God's bringing something up in you that needs some healing, right? And so I especially find that we can sometimes those things happen when we are tired and when we're worn out. So yeah, just kind of thought I'd mention that. And yeah, let me start off class by praying for us today. Let's pray. Lord, I just thank you for each student in this room and each individual here that is a child of yours, Lord, and you created us all in unique and wonderful ways, and you speak to us and you want to heal us through the things that might rise up out of the depths of our souls at times, and through our emotions and through our reactions to things. And I just thank you for that message today, that we could listen to those things, and maybe you can make us a little bit closer to you. You can make us a little bit more whole through those moments, Lord. I pray for the students and their stress this week, in the coming weeks, that you would be with them and give them strength and perseverance, and also give them moments to take a deep breath and relax, and just get a little bit of rest or a little bit of exercise, whatever it is that they need to fill their cups to get them through the rest of the week. And I just pray these things in Jesus' name. Amen. Okay, so I wanted to just discuss a couple things about what's coming up the next 2 weeks in class. So obviously today you got your lab quiz over the muscle function stuff that we kind of talked about what's going to be on that quiz last class. Okay, I want to also point your attention to the lecture quiz this week because it's a little bit different than normal. Okay, it is over. If you click on it, it's over the muscles not covered elsewhere packet. Okay, so what that means is really the main muscles that you learn in this section are the TBL day that you already had, the hip ones, the TBL day on this coming Monday, and then this packet here. Okay, and let me just show you what that is. I have it up on my computer over here. So this is the packet at the end of the notes for this section. Okay, and so this covers all of the lower leg muscles. Okay, this is kind of a view of the lower leg by compartment. Okay, so there's another place where we can kind of group muscles. You guys see that in your packets? Yeah. Okay. Yeah. So I think it's the last one in section 4 there. Yeah. So it's a little ways back. So the purpose of the quiz is really to kind of force you to get into this stuff. Okay, force you to study this stuff, prepare for this quiz, but then also learn these muscles. Okay, these muscles will be just as fair of game for the exam as the other ones that we had a TBL day on. Okay, but this quiz will kind of make you study it. Okay, but this looking, I'll just talk about a couple things with this packet. This looking here is there actually are separate compartments in the lower leg, and you guys saw this in the cat when you dissected it, right? You had to get through those really tough connective tissue membranes, especially on that anterior lateral compartment. So we have the similar thing going on in humans. But one thing we can do with these compartments is we can group them together, and muscles in the same compartment tend to have similar functions, right? And similar actions, right? Just like we talked about with the hip, if it's on one side of the hip or the other, kind of tends to have a certain action. So same idea there. It's a little bit of a way to cut down on the overall memorization, is to group things together. Okay. So it goes through all of these muscles here. And then, it kind of, you can do this grouping here if you'd like. but then it also gets into the forearm muscles. So really lower leg and forearm muscles. Okay, and again, there kind of are compartments to this. There's the anterior side and the posterior side. Anterior side tends to be flexor muscles, flexing of the wrist, flexing of the fingers. Posterior side tend to be extensor muscles, so extending the fingers or extending the wrist. Okay, so those are the ones you need to know. And there are more muscles than these in the body, but these are just the ones you have to know for this class. Okay, so yeah, so all through those forearm muscles. Another opportunity to summarize them, kind of clump them as you learn them. And then just the last couple ones on the abdominal muscles. Okay, we'll see these in the cadavers. Not this week, but I believe you'll start getting into the cadavers next week. And we will see all of these abdominal muscles here. There's really 4 separate ones that you'll see in the human and that you need to know here. So so yeah, we see the rectus abdominis, that's the big one, and the most superficial that we can see that makes like the 6-pack, right? The external and internal obliques, and then the transversus abdominis, those 4 you'll need to know. And so yeah, this is all part of this muscles covered elsewhere packet that you'll want to study for the quiz. And I believe I made that quiz with no time limit. Is that what I said? Yeah. So that's really, if you want to just have it open and have that PowerPoint open and just be learning it as you're taking the quiz, I wouldn't limit your study to only what's on the quiz because you'll need to know them all, but that's a fine way to do it. Just have it open, be learning them, taking, answering the questions along the way. Okay, so that is the setup for the quiz this week. Okay, any questions about that? Okay, all right, let me go back to my iPad here. Okay. All right, so yeah, and then so next week we'll have that TBL day on Monday, as I mentioned, and then the lab quiz will be on the upper. Yeah, maybe today is the first day you guys get into the cadavers. Is that what your professor told you? Yeah, that's what I thought. Okay, all right. I was getting myself confused. Yeah, so you will get to see the lower extremity muscles in the cadavers today. And then and then that quiz next week will be the cat muscles that you dissect this week, as well as the lower extremity human muscles. And yeah, so definitely study those from your packet, as well as you'll get the opportunity to look at the cadavers today in class too. And then exam 4 on Friday next week. Okay, and so this exam is obviously heavy on the muscles, right? So lots and lots of muscles, and most of the questions I try to do with the muscles, you need to really be able to identify the muscle on a diagram, right? Like where is it? If something's pointing to the diagram, which muscle is that? But most of the questions aren't just like, what is this muscle? Most of them are like, which letter is the muscle that does this action or that does this thing or has an origination that's on this side of the hip or something like that, right? So most of them kind of have multi-levels to them versus just trying to identify the muscle to get something at kind of a little bit more of the function of it, as well as the application of the knowledge. So that's usually the way I steer those questions on the muscles. Okay. Yeah, so that's kind of it for that. But any questions about anything coming up in the class? Okay. All right. Okay, well, today we're going to dive into some muscle physiology here. So understanding how muscle contraction works, and really this whole theory, or the understanding of muscle contraction is known as the sliding filament theory. Okay, and so really it's how all those different proteins we talked about last time, how they work together to shorten muscle. Okay, so this background image is from the little video we'll watch, but this is kind of looking at a bunch of myosins, bunch of actins, and kind of how those come together to cause muscle shortening. Okay, so take a sec here, and we talked about all these proteins last time, right? So take a second just with the people around you. Try to remember what the function of each of these might have been. We talked about them a little bit last time, and then yeah, and then take a guess there on the clicker question as well, which in the little GIF there, do you think is tropomyosin? Okay, and the colors are somewhat reflective of what I have there, so give it a shot. So yeah, discuss the function, the part that's covered up with each of these, and then answer that clicker question there as well.
What do you think Zeke is?
I have a question. So I got the new eye clicker from the bookstore and I registered with it
Yeah.
here,
Okay.
but it says that it's still in the base.
So just go, you gotta just change the frequency.
Oh, okay.
So go to AV. Should be ready to go now.
perfect.
Give it a shot. Yep, there you go.
Thank you so much.
Mhm. All right. So go ahead and send that in if you haven't yet. All right. Anybody else? Is that everybody? Okay, so about 90% of you are saying B there. So the blue one, okay? So that is the kind of long string-like one. There's no point in trying to put an arrow to that. I'm not sure why I did that, but the long blue, like stringy one that is kind of going down the whole length of the actin molecule, right? So that's the one that actually covers up the binding site for the myosin head there, right? Okay, questions on that part? Okay, all right, so then let's talk about these different proteins here. Okay, so the heavy chain, the myosin with the head. What did somebody say that the function was there? Somebody raise their hand and tell me what's the function of the myosin or the myosin head? What'd you guys say? You guys talked about it, right? Any thoughts? What does it do? What's the myosin do for us? Josh, what do you think? It helps muscle contraction for sure. Yeah. So this is really the one that moves the actin, right? The myosin head is this guy right here, right? So it is grabbing onto that actin, pulling it right along. Okay. Actin is the one that actually slides when we're talking about the sliding filament theory. It's the one that does the sliding. So in that little GIF up there, it's kind of like the orange one, right? With all the little balls, little beads kind of strung together. Okay, tropomyosin is the one we were just talking about. It's that blue one. So that's what covers up the actin myosin binding site when there's no calcium present. Okay, so it's just kind of in the way of the myosin head binding to actin. Okay, which ion interacts with troponin that we talked about last time? Calcium, there we go. Yeah, so calcium is going to bind to troponin, which changes its shape, right? And then that, since troponin is attached to tropomyosin, when troponin's shape changes, it just pushes tropomyosin out of the way. So now we got an open binding site. All right, so calcium is the one that's going to bind to troponin, like we just talked about. Okay. And then adenosine triphosphate, what this does is actually does what we kind of call cocking the myosin head. Okay, so ATP is our energy source, right? And we would tend to think that we would use our energy source when the muscle actually shortens, but it's actually not that way, right? It actually, the ATP is used when the actin and myosin aren't touching each other yet. And it puts the myosin head kind of into what's called a power pack position. And then it attaches, but the energy's already been used at that point. Okay, but then all that has to happen is for that myosin head to spring back while it's attached to actin, then it pulls it with it. Okay, so it's kind of similar to cocking a pistol, right? So when you put your energy into it is when you pull the hammer back, right? And then all it takes is for that to be released, right? So the actual bullet doesn't come out until the release happens, but you put your energy into it before that. So it's similar to the cocking of the myosin head here. Okay, and both of these interactions, so both calcium interacting with troponin and ATP interacting with the myosin head, which moves that as well, right? Both of those things kind of take us back to some of the early days of this class where we talked about protein levels of structure, right? Remember we talked about the primary level, the secondary, tertiary, quaternary, right? The primary is just all of those covalent bonds that are holding those amino acids together. But then once we get up above that secondary, tertiary, then we're talking about hydrogen bonds, right? Mostly hydrogen bonds, either with the amino acids, how they're interacting with the water around them, or how they're interacting with the amino acid next to it, right? So we got like a positively charged one next to another positively charged one. Those are going to push away from each other, right? Two positive, or a positive charge and a negative charge, those are going to come towards each other, right? All of those little interactions are what give us these perfect shapes of these proteins, right? And then the amazing part about this is troponin has a spot where calcium binds perfectly to it, right? So that's one amino acid that maybe it has a -2 charge or something that allows that negative or that +2 charge of calcium to bind right to it. And when it does that, it totally changes the characteristic of that one amino acid inside of troponin in such a way that the whole thing just folds in a different way, right? It totally changes its shape, moves it, and opens up that actin-myosin binding site for us. And this is what allows contraction, right? Without that mechanism, none of us would be moving, right? So it's this amazing little string of amino acids that are in the exact perfect order to where there's just this perfect little binding site for a calcium. And when it binds to it, totally changes the shape of that thing. And this is what allows us to contract and relax muscle, right? Same thing with ATP. When ATP binds to the myosin head, that phosphate goes on to the myosin. And by that phosphate binding to myosin, again, completely changes the shape of that myosin, right? So it's binding to some amino acid on there. I don't know biochemistry well enough to be able to dissect that with you. But it binds to some amino acid, which completely changes the shape of that myosin head. It cocks it, right? And then it can attach to the actin. As soon as that phosphate comes off, it goes back to its original shape. But as it's doing that, it pulls actin along with it, right? So it's just kind of the amazing part of how these proteins are just designed in such a perfect way. Just one little thing binding to it is really what animates all of life in biology, right? And just all creatures move because of this mechanism right here. And it just has to do with this perfect little placement of all those little amino acids. So, okay. All right, I get excited about that stuff, but let's talk about this. Okay, so we're going to walk through the details of this a little bit today. So there's really two different processes that we're going to talk about. First one is called excitation contraction coupling. This is going to start with what's called an action potential. We'll get into the details of what an action potential is later, but it's really a nerve signal. Okay, so an electrical signal traveling down a nerve to activate some kind of tissue, in this case, a muscle cell. Okay, it starts with that, and it ends with that part we've already been talking about, which is calcium binding to troponin. Okay, so there's a bunch of little steps in there in between. So this is really like preparation for contraction. Okay, so preparing for contraction. Okay, so that's kind of our first part. And then second part is once we're all set up and ready for contraction, then we move into what's called cross-bridge cycling. And cross-bridge cycling is really what this little GIF was showing us. That's pretty much cross-bridge cycling. We'll look at it in a little bit more detail of a format showing what's going on there with the myosin head and the ATP and the calcium and all those things. But that is really cross-bridge cycling. So that is actually the muscle shortening. Okay. All right, so let's start with the excitation contraction coupling one. And so with both of these, we kind of have these steps in this process. And the way that we'll do this is I'm going to show a little video here that kind of walks us through these steps, through some animations. And then I'll kind of talk through these steps, but the slides that follow this kind of walk us through these steps as well. So you guys can kind of use the steps from the previous one, talk about what's going on in each of these images here, kind of fill in what's happening. Okay, and then we'll move over to the next one, cross bridge cycling. But let's just start with excitation contraction coupling first. And this one, the video shows both, but we'll just work on filling in the excitation contraction coupling one first. Okay, so I'm not necessarily going to read these right here, but I will start the video and then we can go from there. So let's go over to that here real quick. It's got super cool music too.
The actual potential down that way. You can see it changes shape on the bottom. They're now open and active.
Yeah.
And let's do this on your clickers while you're talking through this would be send in the number from the first slide that we number or numbers that would be happening here on this slide here. Okay. Just send in one number, but yeah, just put in one. There's more than one going on here, but put in one. All right, go ahead and send that in if you haven't yet. All right, good. Looks like most of you here. Okay, so we are between 4, 5, and 6, which is good because those are the ones that are all occurring here. But let's start back with the first one here. Okay, so this is a axon. Right, so axon is a cellular extension that comes off of neurons, right? Our nerve cells. And so it's a motor neuron, right? A motor neuron is a nerve cell that is controlling our skeletal muscle, okay? Or it's really sending a signal from the central nervous system out to the periphery, okay? And so we have one little branch of that coming down to a single muscle cell here, and that's where we're zoomed in on, okay? So we get that electrical signal traveling down this way, okay? We'll talk actually a little bit more about a little bit more detail about some of the events that cause this release here, but it's going to release a neurotransmitter. Okay, well, but that's what we'll get into more detail on the next couple of weeks. And that neurotransmitter pretty much causes an action potential here. And a lot of times we'll just abbreviate action potentials as AP. Okay, but that action potential pretty much spreads from the neuron to the muscle cell. Okay, I'll explain the details more in there once we get into the nervous system part, and then we talk about the neuromuscular junction. But that's the first step, right? So arrives at the neuromuscular junction, starts an action potential on the muscle cell, right? So we got steps really 1, 2, 3 occurring there on the first one. Okay, and so we see that action potential traveling down, right? And we can start to see this structure here, which lets the action potential dive deep. What did we call that guy again? This one right here? T-tubule, right? Yeah, absolutely. So those are the T-tubules. So now we can see that T-tubule a little bit closer here. We see the action potential diving down the T-tubule. Okay. And yeah, so we got that part. And then we move over to this one. Again, here's our T-tubule. Action potential is diving down. We can see that there's these special little connecting points, which are little proteins that connect that T-tubule over to a calcium channel on the sarcoplasmic reticulum, right? That's what this is. So calcium channel here, this is that sarcoplasmic reticulum. This is where all the calcium is stored inside of skeletal muscle. Okay, so that action potential changes the shape of that protein that's attached there, which then opens up the calcium channel, and then we start dumping calcium into the muscle cell. And if you kind of look closely back here, you can kind of see the actin, the myosin. So the sarcomeres are all just right back there behind all of this, right? So calcium is being dumped into that space. So yeah, I think we're seeing all 4, 5, and 6 here from the previous or from that initial slide. Okay, so we're releasing calcium here. Okay, once we release calcium, now we're zooming in on the sarcomere a little bit more. Again, we can see the calcium releases, just showing that a little bit closer, showing closed versus open. Okay, but now we see troponin without calcium bound to it. And then here's troponin with calcium bound to it, right? So that calcium binds to it, changes its shape, moves, and then it pulls tropomyosin along with it when it moves. Now we've got these open binding sites ready for that myosin head to attach to. Okay. Questions on that? Okay, so that's our excitation contraction coupling. This is getting us ready for muscle contraction. Okay, and now we'll talk a little bit about cross-bridge cycling. Okay, with a cycle like this, we can really start at multiple points, but this, yeah, so it's just a choice of like exactly where we're starting. So ATP hydrolysis, this is breaking down ATP. It's the reaction that's going to break down ATP into ADP and phosphate. The P with the I there is just called inorganic phosphate. Okay, so ATP is when it has 3 phosphates on it, right? ADP is when it has 2. Okay, so we've just taken one of those phosphates off. Again, this phosphate is going to bind to myosin, and it's going to change the shape of that myosin head, like we talked about. Okay, so it's going to cock that myosin head, then it's going to go ahead and attach to actin. Okay, and then once, and the video was a little bit not exactly perfect on this part, but I still like that video the best as far as visualizing things. But when ADP and phosphate are removed, are released from it, that's what causes the myosin head to go back to its original position, which is called the power stroke. Okay, and then we only can detach that myosin head from actin when we got a new ATP that comes in. Okay, so a new ATP comes in, so new energy, that attaches to myosin, and then we can detach those two. And this has a pretty significant application. If you've heard of rigor mortis, right? When after somebody has died and their muscles are all stiff, right? This is why, because all the myosin heads are attached to actin. There's no new ATP being generated because the person's not breathing anymore, right? And so we don't have new ATP. So we got the myosin heads stuck to actin all throughout their muscle, and that's why those muscles are stiff, right? And that's what causes rigor mortis. Okay, so take a maybe just a minute or so with the people around you and work through these images here. Okay, and then it kind of shows it in the cycle format here too. And then I'll have you go ahead and answer this question here on your clickers when you get through that. Okay, so walk through each of those slides, talk through what's going on, and then answer that clicker question there at the end. Yeah, that's when the muscle actually shortens. Yeah, but really it's referring to the myosin head, like when it kind of springs back into its original position. Yeah. All right, so you're walking through these slides, talking about what's going on, and then go ahead and when you get to it, send in the answer to this question here. Which one of these letters here is the same as the step 1 from the previous slides? All right, maybe about seven or eight more of you still to send that in. Anybody else? Go ahead and everybody just press a number again. We're still missing like 4 or 5, it looks like. Or sorry, press a letter again. Okay, there we go. That looks better. All right, so about 2/3 of you are saying C there. Let's go. We'll start with the first slide though. We'll come back to that. So let's cross-bridge cycling begins here. Okay, so this first step again is that cocking of the myosin head, right? You'll notice that these two are not attached yet, right? And again, this is when we use our energy. This is the equivalent of pulling that hammer back on a gun, right? You're putting your energy in, but there's nothing actually happening yet, right? So the energy from the ATP is being spent, right? And really, again, that's just because this phosphate binds to a specific amino acid that changes the whole shape of the protein, right? So it puts it into its cocked position. Okay, so that's the first one, right? And now those two can attach to each other, right? So we see the myosin head and the actin now attaching to each other. Okay, we see ADP and phosphate are still there. Okay, and then the power stroke is initiated by these ADP and phosphate coming off, right? And by doing that, when that phosphate comes off, it was attached to the myosin head. It was changing the shape of it, right? So now it can go back to its original shape, which then it just pulls actin along with it since it's attached. Okay, and then we see that last step with a new ATP that just came. That's what allows for the disconnection there between the actin and the myosin. And we can start the cycle over again. Okay, so step 1 of this one was when we're cocking the myosin head. Okay, so in this case, most of you said C. So that looks like when we are going from a full ATP here to a broken down ATP, and we can see the movement of the myosin head going in that direction to be into what's called the power packed position. Okay, so that is definitely correct. That would be number one from the previous slide. Okay, so we can see all those things happen in order, and it just kind of happens over and over and over again, literally millions of times when we're moving our muscles around, right? Yeah. Questions on any of this? Okay. All right. And then this slide just kind of nicely puts those two things together, right? So we had to start with excitation contraction coupling first, right? So we had to get that signal from the neuron right here. We had to make that action potential happen on the muscle cell, right? That had to go down and cause calcium to be released. Okay, we got calcium to be released. It had to go bind to troponin, move tropomyosin out of the way, and then start that whole chain of events that is cross-bridge cycling. Okay. In a couple weeks, over the next couple of weeks, we'll backtrack a little bit. We'll talk about the events here at the neuromuscular junction. Okay. That's kind of what actually happens to start that action potential on the muscle cell. And then we'll backtrack even more when we get to the nervous system, talk about how action potentials happen, talk about the parts of the brain that are used to activate these motor neurons, and kind of put all that together. Okay. All right. Well, just a last bit here that we're going to talk about. Oh, that thing got moved. Did all these get moved? Okay, good. Sorry, that one. Okay, well, you can see what it is there. But what I wanted to do here, and I know you guys kind of walked through this in lab already, with taking a step, and yeah, and one part about that, Professor McGaw was texting me a bunch during lab. He was, I don't know, some people were struggling with especially like the little bit of extension that happens with the knee while you're doing that push off phase. There's actually around like 12 phases to walking. So we just kind of put it into like 2 or 3 phases, right? So definitely like a simplified approach to that. But if we start here with the weight bearing leg, right? And this isn't, I don't know if you guys even talked about the weight bearing leg in lab. I think you were just focused on the other one. But whenever we're taking a step, right? So we have my left leg is down. I'm going to swing my right foot forward. It's obviously important here. You can see what that one is to stabilize our pelvis of the leg that's weight bearing, right? As we're swinging our foot forward. And again, we talked about the gluteus medius having this action before. But as I lift my right foot up, if my gluteus medius wasn't contracted, my hip would fall down like that, right? Because that's actually connecting my femur up to my pelvis, right? It's holding me stable there, right? So it could kind of fall over like this. So we kind of just have this really like an isometric contraction of the gluteus medius happening there. Isometric meaning that it's not moving, right? It's just stabilizing, right? It's just holding it, right? And we have a lot of muscles that help us in this way. Okay, so that's just kind of the simple one there. But then the other one, the other leg, right? So we've got the stepping forward leg. Okay, so we've got the push off part and the swing part. Okay, so during the push off, we see a few things going on. Okay, so we want to look at each joint. So we want to look at the ankle, the hip, and the knee. Okay, so with these motions here, what I want you guys to talk about for a second would be which muscles lead to those actions. Okay, so talk with each other on each of these. Which muscles will you use for plantar flexion, for the push off, right? And just work your way down through all of them. We'll come back together here in a couple minutes and just check in on that, and we'll be done. Go ahead.
So, plantar flexion, ankle extension. I remember this like I was like extension. So it's pushing the back. So well, I think.
And then
Yeah. And then would that also be the what's called or the if I had and for like 30 seconds.
Oh, that long one, yeah.
You've had fun playing with it last night.
Yeah.
You were writing some stuff on her arm or something? Is that what that is? She's like, Miss McKenna's friend was drawing on me. That's right. I was like, that's awesome. Yeah, she had fun running, playing with you guys. Yeah, that's really fun. All right, guys, let's go ahead and come back together here. Okay, so let's talk about the first part. So if we're thinking about this, our right leg being back, we're going to have to push off first, right? And the knee with this is complicated because the knee does multiple things, right? At the very beginning, there's a little bit of flexion, but then there's definitely some extension too, right? So you just don't want to get too caught up in trying to figure that out because like I said, there's like 12 different stages, right? So we're just kind of choosing to talk about the knee flexing at this point, and then it's going to extend when we kick it out, right? That one's pretty obvious, but when it's back here, it's a little bit different, right? There's a little bit of flexion, a little bit of extension happening. But let's start with plantar flexion, right? So it's pushing down with our ankle there. So what are our big plantar flexors? Gastrocnemius. What else? Soleus. Tibialis. Which one?
Anterior.
No, not tibialis anterior. We'll see that one maybe down below. But yeah, the two big ones, soleus and gastrocnemius. Okay. If we could also put in fibularis longus. It's also a plantar flexor. And the way you know this with the we haven't done the ankle muscles yet, but that's part of the muscles covered elsewhere packet. But if we look at the ankle and we kind of put an axis right through our lateral malleolus, right? Like take like a picture, like a pencil going right through our lateral malleolus. If a muscle passes behind that, posterior to that, then it's a plantar flexor. If it passes in front of that, it's a dorsiflexor, right? Because that's the axis of rotation, right? So depending on which side we look at, same thing with inversion and eversion, right? We can kind of picture a line going down straight down through our foot. If it inserts on the medial side of that, it's going to be an inverter. If it inserts on the lateral side of that, it's going to be an everter, right? That's just a way to kind of picture it. And with a lot of this stuff, it's much easier to understand it than it is to memorize it. Right? So you could go through and memorize every single little muscle, try to get every single little origin insertion, and then probably still miss a question that I ask you about, like, is this one an inverter or everter, right? But if you could just picture the muscle and think, oh, it goes to the right, to the lateral side, it's going to pull that way. Obviously, it's an everter, right? That's so much easier than just trying to like smash it all into your brain and hopefully it comes out at the right time, right? So if you can kind of take a step back and just try to understand in those ways, it's super, super helpful. Okay, hip extension. So our hip is going back a little bit, right? What are our big hip extensors? What'd you say? Hamstrings. Okay, what else? Glutes, right? Hamstrings and glutes for sure. Yeah, so hamstrings, got 3 of them there. What's the one on the far lateral side? Biceps femoris. There we go. Okay, it's got 2 heads. You'll see the 2 heads in the cadavers today. If your instructor points those out, if not, then say, hey, can we see the other head too? Because I used to always point it out. Okay, so that one, and then we move our way in. Go ahead. That's what I was just kind of talking about, how it's like very complicated and there's multiple things going on. For what? For the quiz? You're not gonna have to worry about that for the quiz. There you go. That solves that. Okay. Don't normally get those questions like that answered, like what's gonna be on the quiz, but figured it'd make your life a little easier today. Don't worry about that for the quiz. Okay. All right. Okay, so if we're going in from there, so we got biceps femoris, then we move to the middle one. What's the hamstring in the middle? Tendinosus, and then all the way medial. Membranosus, good. Okay, so we got those three. We could definitely add in the glutes. With a question like this on the exam, as long as you put in a muscle that does that, fine. I don't have the glutes here, but if you added a glute muscle on a question like this, that's fine. If it does that action, that's totally fine. Okay. We tend to use our glutes more when our hip is flexed a little bit more, like we're going upstairs, right? So we come up and then that's where we kind of hit our glutes more. When we're here, it's going to be more hamstrings, but if it does that action, you'll get it right. Okay. A little bit of knee flexion, also hamstrings, and then gastrocnemius also helps us with this too, right? Because remember that we haven't really learned it yet, but gastrocnemius crosses the knee as well as the ankle. Okay, so it has a medial head and a lateral head, and it goes up over the back of the knee. So it's also a knee flexion. Okay, so hip flexion. Now we're going to start swinging that leg forward. Okay, hip flexion. What's our primary hip flexor? Iliopsoas. Okay, what's another one? Pectineus. There we go. What's one more? Rectus femoris. There we go. So we got all three of those there. Okay. Our knee's going to extend now. What big group does that? Knee extension? Quads do. Okay. The one on the very top, we just said it from the last one. Rectus femoris, right? And then we got the medial one. Vastus medialis. Middle one, vastus intermedius. Lateral one, vastus lateralis. Okay, so on a question like this, you wouldn't want to just write quads, right? You want to write the names of those muscles. Okay, and then ankle dorsiflexion, we're pulling our toes up towards our shin. Isaiah, get us here. There we go, tibialis anterior. Okay, and then how do we get our toes lifted up too? Yeah, so then we got our extensor digitorum longus as well, right? And that one, since it crosses over the anterior side of the ankle, it's going to pull up that foot a little bit too, as it also pulls the toes up. Okay. All right, leave that up there for a sec, but we're all done. Have a great lab today, and we'll see you guys on Friday. Yeah.
Can you show me the part? I really need to.